Okay, hi everybody. It's 4 afternoon in the east coast of Australia at the moment, 9 a.m. in Israel. Um, Let me first acknowledge the Jagera and Turrbal people. They are the traditional custodians of the land on which we gather here today. That's in Brisbane, Australia. We pay respects to their elders, past and present. We extend that respect to Aboriginal and Torres Strait Islanders people here today in Australia. That was our acknowledgement of country. And now it is a great pleasure to introduce Dr. Eddie Citrin. He runs the Citrin Lab at the Volcani Institute. So that's a leading research institute for agriculture in Israel. We've had another Volcani Institute member speak previously at the Australia-Israel Scholar Seminar Series. And we are very glad to have Eddie's talk. So without further ado, it's over to you. Thanks, Eddie.
Hey, thank you, Yoni, and thanks everybody for joining. And I'd like to give a brief overview of research in my lab over the past, I would say, 14 years that's been focusing on antibiotic resistance in agro-environments. I think most of us are familiar with Alexander Fleming, the discovery of penicillin, which was followed by discoveries by Selman Waxman, many other antibiotic compounds. And this led to what was called the antibiotic era, where by the early 1970s, there was a feeling that we had defeated infectious diseases, and indeed, public health authorities felt that we could move on to other problems like heart disease and cancer. And we had antibiotics. Every single bacteria that caused infectious diseases was treatable, and therefore we had basically solved the problem. If we move along to the 21st century, we've actually arrived at what's been coined the post-antibiotic era. And this citation is from Margaret Chan, who was the former Director General of the World Health Organization, who said that a post-antibiotic era means, in effect, an end to modern medicine as we know it. Things as common as strep throat or a child's scratched knee could once again kill. And if we look at projections, and this is a projection of deaths associated with antibiotic resistance in the year 2050, we're talking about close to 10 million deaths annually. And as you can see, most of them are predicted to be in developing regions such as Southeast Asia and in Africa. And the issue with antibiotic resistance, as many of us maybe know, is the fact of selection, where the more we use an antibiotic or an antimicrobial, the more the ratio of resistant versus sensitive bacteria will increase. And one of the other significant factors for driving antibiotic resistance is what we call horizontal gene transfer, where genes can actually be transferred from one bacteria to the other. They don't necessarily have to be closely related. Often what's known as conjugation is the most significant driver of horizontal gene transfer. And when we have this transfer of plasmids of these genetic elements from one bacteria to the other, we're not only talking about one gene, we're often talking about plasmids that harbor up to 10 or even more genes. And this has led to a situation which has been coined bad bugs, no drugs. So basically a bacteria that is multi-drug resistant or even pan-resistant, basically resistant to everything we can throw at it, is untreatable. Now, I think most of us are familiar with the issue of antibiotic resistance in clinical settings. But over the past years, people are talking more and more about what's known as One Health, where we have this very, very intricate association between human health, but also animal health and ecosystems conditions or environmental health. And often there's basically a very strong interface between these different environments. And if we specifically look at agriculture and things that I've been dealing with over the past 10, 15 years, we know that, for example, in livestock agriculture, and agriculture is there's actually more use of antibiotics than in human use. So of course, this is causing selection. We have transfer through meat, through fertilization, through aerial transmission to humans. And my lab has published a number of studies on both poultry and aquaculture. But what I'll be focusing on today is this interface between wastewater, and if we're talking about developing regions, this can often be untreated, so basically raw sewage, or in more developed regions, treated wastewater, and the environment, and of course, through food webs or water cycles, we're getting back into the community, and this, of course, is causing this integration with human health as well. So we know that in Israel, we recycle a lot of our wastewater. This is something that started in the mid-1960s. And as you can see, we've crossed the 50%. So over half of the irrigation water in Israel is treated wastewater. And over 90% of all the treated wastewater that's produced in Israel is used for irrigation. And of course, this is very, very significant. But the phenomenon that we see in Israel and also in certain parts of Australia, as we can see, is growing globally due to increasing global populations, and of course, climate change, which is causing more severe droughts, which necessitates irrigation. So this is a local problem in Israel and perhaps in South Australia, which is growing to become more of a global problem. And of course, we need these water resources for irrigation, for food security. So my working hypotheses are basically two. One is that consumption of crops irrigated with treated wastewater basically transfers antibiotic-resistant pathogens to the gut, which can cause disease. So we have this direct connection between antibiotic-resistant pathogens coming from the wastewater that can potentially be transferred to humans through consumption of produce. And the second issue is that following consumption of crops irrigated with treated wastewater, antibiotic resistance genes are transmitted to the gut where they can be horizontally transferred to pathogens. So this is an indirect connection where, of course, these genes, upon transfer to pathogens, are making the pathogens much more hazardous because, as I mentioned before, they are much more difficult to treat due to antibiotic resistance. I'm going to give a number of very, very brief overviews of studies that have been done in the lab. This was done by a PhD student, former PhD student, Roberto Morano, published in Water Research. And here we used a culture-independent approach where we were looking at a myriad of antibiotic resistance genes using quantitative PCR analysis. And basically, we're looking at a continuum from the irrigation water itself, which often went through reservoirs, sometimes was directly used, looking at the soils that were irrigated with this water and also at produce. And again, quantifying genes that can be indicators for antibiotic resistance in the environment. And very, very briefly, this is kind of a summary of what we saw in effluent water that was used for irrigation, and in corresponding freshwater samples. We didn't always have parallel freshwater irrigated crops, but in some instances we did. And what we see when we look at these different genes is that in freshwater, levels were generally very, very low, or not detected, or detected but not quantifiable. Versus in the effluent samples where we see much, much higher loads of these antibiotic resistance genes. And if we kind of extrapolate this data to daily loads of antibiotic resistance genes entering the soil in copies per meter square per day, we're talking about very, very, very high levels of resistance. However, if we look at the soil and the crops that are being irrigated with this water, we see that in most cases, gene levels are either below detection limits or detectable but not quantifiable. And there was no real correlation between the gene loads that we saw in the water and in the corresponding irrigated soils or crops. So our conclusion from this was that soil is an ecological barrier that impedes dissemination of antibiotic resistant bacteria and antibiotic resistance genes. This was also the conclusion from a previous study that we published. And to try to validate this hypothesis, Roberto, along with Tamar, who was a master's student, and Chetty, who was a postdoc, did these microcosm experiments where we basically spiked these multi-drug resistant bacteria that we could actually fish out from these soil microcosms. So it was a one-time spiking at time 0. And then these soil microcosms were incubated, kept at a fixed water content. And we looked at the abundance of these genes over time, and what we can see is that indeed, when we look over time between 8 and 28 days after the initial inoculation with these multi-drug resistant pathogens, we could see that they completely disappeared. And basically the conclusion indeed indicated that the soil is a barrier to these. However, when we inoculated all these soils in a rich, what we call copiotrophic medium, and looked for these bacteria, we saw that with the exception of this sample, which disappeared, the pathogens disappeared after 8 days, all the rest of the samples basically recovered, indicating that these bacteria were present, they were just below detection limits. So we went on to do a very, very intricate experiment. This was followed up by a guy, Davidovich, who is presently a PhD student in the lab. And basically, we used this kind of under the radar hypothesis, looking at lettuce that was irrigated with either treated wastewater, which was very, very low quality, fresh water, or amended with manure, which is often used as a biological fertilizer. I won't go into all the methodology. I'm just going to give a little bit of taste of the results. So the first conclusion was that if we look at rhizosphere, so the lettuce root samples versus the phyllosphere, which are the lettuce leaf samples, we see that the diversity of the microbial community was much, much higher in the roots than in the leaves. But when we did these enrichments, and I didn't mention, but we enriched in both aerobic and anaerobic conditions because the human gut has both areas that are aerated and areas that are anaerobic. We see all the enrichments showed a significant decrease in diversity. As a microbiologist, this is actually very bad because we're causing a bias. We're only focusing on those fast-growing bacteria that like rich media. And you can actually see this also by looking at the microbial community. But when we look at the actual populations that were induced under these nutrient-rich enrichments, and this is the leaf, the roots, sorry, we see that in the freshwater enrichments, we see mostly bacillus. So this is basically generally a non-pathogenic group of bacteria, and they dominated these enrichments in the freshwater irrigated root samples. But when we looked at the manure-amended root samples that were enriched, we do see bacillus, but we also start to see emergence of pathogen-associated taxa, like Enterobacter and Acinetobacter. And when we look at the treated wastewater irrigated enriched samples, we see very, very low abundances of bacillus, and we start to see higher abundances of Aeromonas, Escherichia, Klebsiella, and again, the same Acinetobacter. So again, a much, much higher ratio of these potentially pathogenic bacteria. So on the one hand, we're creating a bias and focusing on specific groups, but on the other hand, we believe that we're enhancing those groups that are clinically relevant. And we see a similar trend in the leaves, but much more chaotic. So we believe that in the roots, we have this stochastic kind of enrichment of groups that are pathogen associated. In the leaves, it's much more complicated, but we do see a much higher relative abundance of these pathogen-associated groups in the enriched manure-amended and treated wastewater irrigated samples. Now, here we're specifically looking at antibiotic resistance genes. And again, I won't go into all the details, but we see in the roots the freshwater irrigated samples, we hardly see any of these clinically relevant antibiotic resistance genes. These genes are much more abundant in the manure amended samples, and real complete domination from both abundance and the diversity of these genes in the treated wastewater irrigated enriched samples. And again, a similar trend, less pronounced, but a similar trend is seen also in the lettuce samples. And we see a similar trend as well when we did these isolation of specific E. coli bacteria. And interestingly, when we looked at the potential of these multi-drug resistant E. coli that were from the enriched samples to transfer plasmids to a model sensitive recipient strain, we see they have the capacity to transfer resistance. So not only do we see higher levels of pathogen-associated bacteria, we also see the capacity of these bacteria to transfer their plasmids to sensitive bacteria. So to kind of summarize the bottom line of these experiments, when we enrich these samples, we see a significant proliferation of bacillus. However, if we go from the more pristine freshwater irrigated to the more anthropogenic treated wastewater irrigated or manure amended samples, we see proliferation of Paraclostridium under anoxic conditions, Acinetobacter under aerobic conditions, and under both aerobic and anaerobic conditions, Enterococcus, Escherichia, Aeromonas, various Enterobacteriaceae. I won't go into all the details. We see a huge array of antibiotic resistant bacteria and antibiotic resistance genes. And interestingly, these include several of the most alarming antibiotic resistance threats characterized by the US Center for Disease Control. So again, to summarize, these under the radar enrichment methods create significant bias, but they do allow us to see potential pathogens and antibiotic resistance genes that are associated with anthropogenic processes in agriculture. And just looking forward, future directions that we're currently involved in, I have a project now in collaboration with Kerry Hamilton, who's a professor at Arizona State University. Kerry is an expert in quantitative microbial risk assessment, and what we're trying to do is apply our under-the-radar tools to link into their quantitative microbial risk assessment models in order to assess risks associated with treated wastewater irrigation. Of course, Arizona is also a place that significantly uses recycled water for irrigation, and therefore we want to look at the risks of how this is affecting antimicrobial resistance. Another project that's actually also in collaboration with a number of groups, including Stephen Georgovich from the University of Technology in Sydney, which is being led by a colleague of mine, Sefi Warnick, from the Agricultural Engineering Institute, where Yafit came from, is to develop bioelectronic-based chips for rapid detection of antimicrobial resistant bacteria and antibiotic resistance genes. And again, we're using our knowledge from the field to design specific probes which could be used for these bioelectronic chips. So with that, I'll finish, acknowledge, of course, all the students and postdocs, Judith, my lab manager, and the collaborators from various institutes, funding agencies, and of course, if there are any questions, I'd be very, very happy to answer. Thank you very much.
Thank you very much, Eddie. I will clap in the name of others. Maybe some others can find the correct button. It was an intriguing talk, and I love the combination of applied research and fundamental research that you do at Volcani Institute. Are there any questions?
Yoni, may I ask some? It's Ross.
All yours, Ross. Yes.
Yeah. Do you want to see my face, or are you happy just to have my voice?
Happier with face, but voice is good.
Oh, there you go. You can have a quick. The sunglasses are because I have eye problems with screen time. It's not because I want to maintain anonymity. I enjoyed that talk very much, Citrin.
Thank you.
So I was a director of the biotechnology teaching program at my university for nearly 2 decades. So I've worked a lot in diagnostics and broad spectrum bacterial detection. I just had a question about the clay loam soil that you talked about in your pilot experiments. Was that sterile, or was it pre-sterilized, or was there an endogenous microbiome in that soil?
Yeah, so the soil was not sterilized. So basically our hypothesis was, and this is based also on a study by a colleague here, Dror Mintz's lab, where they actually showed that when you irrigate with treated wastewater, the water-associated microbiome generally cannot persist due to persistence. So in ecology, this could be called resilience or resistance of the native community. So we specifically wanted to use soils with their indigenous communities to see how this E. coli, these 2 E. coli strains could survive over time.
Okay, absolutely, because maybe my first thought was that the endogenous microbiome might be contributing, and you would have thought of this, contributing in a very important way to the ecological barrier to these pathogens. So,
yes, so in fact, I'm now submitting a grant along with somebody named Mike Williams, who's from CSIRO in Adelaide, where we're trying to look at various what we call nature-based solutions to kind of exploit these environmental, robust environmental microbiomes to reduce both chemical and biological contamination coming from wastewater. So
okay, and I'll just ask a quick follow-up question. I can see others wanting to ask questions as well. In the larger scale irrigation experiments, what happens if you use sterile water with the irrigation and then do the enrichment experiments?
So that's not something that we've actually done, but it's an interesting question. But again, I think from similar studies that we've done with compost, it seems that the capacity of, if you have a robust environmental community, the capacity of like an invading community to persist is generally quite low. However, this might change if the intensity or the load of what's coming in passes a certain threshold. Again, these are things that we've thought about in theory, but haven't actually done the experiments. It's something we want to do, but the question is, when you get to a state of what we can call dysbiosis, where the indigenous or local community can no longer basically maintain this capacity, and then it's maybe overrun. So this is theoretical.
I was just thinking more in terms of fundamental control, what you get out of what you enrich if you basically don't have any contaminant organisms, whether there are pathogens already hiding away in that soil that weren't introduced in the medium.
Yeah. So that's a good question. Actually, there's a colleague from Germany who's been doing a lot of work on that, and then I think it kind of focuses on how you define a pathogen. For example, Enterobacter can be a potential an opportunistic pathogen in certain cases in humans, but it's actually a group that's known to persist very well and often is even associated with beneficial traits in plant roots and leaves. So I guess it depends on what you call it.
There's well-known ones like things like Strep pyogenes A, for example, that are notorious for infecting landscape workers or gardeners. They get a scratch and they can be very quickly. In fact, it happened to my son himself. Ended up in intensive care from a Strep pyogenes, just from a soil scratch when he was gardening. So there can be some interesting things lurking even when you're not irrigating with wastewater. But that was a very stimulating presentation. Thanks, Citron. So.
Thank you very much.
Thanks, Ross. We have a question in the chat. I'll read it out. And the question is, how do you expect an introduction of chimera drugs to affect these trends in propagating these drug-resistant genes? Would this just be a temporary solution until new bacteria evolve against it?
So the answer is generally yes. So I didn't have time here, but there's a very, very nice timeline showing the top of the timeline shows when antimicrobials were introduced, and the bottom shows when resistance was first identified in community in clinics. And often you see development of even synthetic or semi-synthetic drugs where the developers will claim that they are extremely difficult for resistance development, and then after sometimes less than a year, you see resistance. So there are basically two trends of thought. I think the truth is somewhere in the middle. One is that resistance is a default, and therefore we're in this type of arms race where we have to just continually develop new antimicrobials, and once there's resistance, then go to the next. But I do think there's a very, very strong correlation between use and resistance, and therefore, if we wisely use antibiotics, and I think it's not only not using antimicrobials when they're not needed, but also sort of dictating which drugs can be used for different conditions. Obviously, you don't want to use last resort antibiotics for things that you don't need them for. And the less use, the less selection, and obviously we might see an increase over time in resistance, but it's going to be much slower. So I think we have to continue discovery of new antimicrobials or various approaches that aren't necessarily antimicrobials, but things like phage therapy, but also really, really think about how we're using these drugs and be very careful. So it's sort of a combined effort.
Gotcha. Thank you. Yeah, and maybe I'll close with a question. It's a bit more of a kind of an outsider question. I come from the world of statistics and mathematics, and looking at your work, it seems like, of course, there's a lot of experimental design and hardcore statistics and perhaps some machine learning. My question is, do you also try to think of things mechanistically from a scientific perspective? So, for example, that hypothesis you had about soil, try to model things from first principles and see what is the reason, etc. How do you balance the stats and the mechanistic first principles modeling?
So yeah, it's an excellent question, and I think, and this goes back to one of the quotations from one of my postdoctoral advisors, but that the trend basically was that we can't really address biological questions unless we have very, very strong, hypothesis-driven questions. So basically, a lot of the work that we do is using sophisticated bioinformatic platforms, statistics, etc., which the students today actually know how to use much better than I do. But I really try to push them to understand the basic questions that they're dealing with, and to try to understand and first go into them. And sometimes I think I also probably don't ask the basic questions as much as we should, but I think one thing that came up from these enrichment studies is the fact that when we talk about antimicrobial resistance, maybe many things in biology, it's not a black and white situation. And they're basically processes that are very, very stochastic. You know, these are random effects that could be caused by all sorts of different processes that are very, very hard to pinpoint versus things that are deterministic. So one of the things that I think we're very interested in now is when we try to put our data into risk assessment models, how do we determine which things are really deterministic or really based on the irrigation water or based on the processes that we're actually looking at versus things that can be random. And this is something that I think we need modelers and people that can help us maybe plan the experiments better in order to be able to test these models empirically. I don't know if I answered your question.
Yeah. You certainly gave a direction. Intriguing. Yeah, on that note, I want to thank you again, Eddie, for taking the time and sharing your work. You said in our chat prior that at least you plan to be in Australia 2 years from now for a conference. So let's hope to intersect then face to face and have plenty of collaborations prior.
That would be excellent. In fact, I just 2 weeks ago returned from there's a conference series called EDAR, which stands for Environmental Dimensions of Antimicrobial Resistance. So the last one was in Montreal, and they passed the torch over to the Australians. The next one is going to be in 2 years in Brisbane. But
Wonderful. Yeah, that's a small period, that short period on the academic timeline. Okay,
right.
so the listeners can find you on our website and elsewhere, and we'll be in touch. Thanks so much.
Thank you. Thanks for initiating these meetings, and thanks for inviting.
Bye-bye.
Bye-bye.
Bye, everyone. Thank you.